Okay, so welcome to your official lesson on the Krebs cycle. Okay, our goal here is to take the acetyl-CoA that we've just created when we oxidize pyruvate, and we're going to chop it all the way down so that the original carbons from glucose are gone. We have 2 carbons left from the original glucose, so I can expect that I will make 2 carbon dioxides. I already made 1 carbon dioxide during the oxidation stage of pyruvate, and now, so I lost 1 there from my 3 carbon pyruvate. Now I'm going to lose 2 more. So for sure, we're going to produce a couple, every time we go around Krebs cycle, we produce a couple of these carbon dioxides. Going back to the oxidation step though, I've told you that when the carbon dioxide got knocked off here, we stole electrons and made an NADH, right? We'll take a look what happens in the Krebs cycle when we knock off carbon dioxide. So on the right-hand side there, you can see we're going to be chopping off a CO2, and what do you know? We're stealing electrons and making an NADH. And then we do it again, and what do you know? That's our last carbon that existed from the original structure, from the original glucose. Last carbon to cut off, what do you know? I make another NADH. So in the first 3 instances of NADHs being made from a pyruvate, you always make CO2. However, we're also going to steal electrons down here at the bottom, the FADH2, on step 6 here. This FADH2 is also an energy carrier. It's also an electron carrier. Does exactly the same type of an idea of what NAD does, steals electrons. It's a little bit of a lower level version of energy. It'll interact a little bit differently with our membrane when we're delivering the payload of electrons. But there you go. We still some more electrons at the end here. And then finally, one more time right up at the top. Between malate and oxaloacetate in step 8 up there, you can see we can steal one more time. We have just enough energy to steal those electrons before we reboot the cycle. Oxaloacetate. We have already seen oxaloacetate. And if you look down to the left, we've already seen malate as well. We saw it about 20 seconds ago for about 20 milliseconds when we talked about C4. So I'm just going to pop back to our photosynthesis conversation and show you where you already saw them and show you the little group that they belong to so you can understand that, again, we are using the same molecules over and over and over again. So let's just pop back briefly to photosynthesis. This is our C4 photosynthesis conversation where instead of the carbon dioxide being immediately attached to rubip so that I can make what I need to make my 3-PGA, and then it's 1,3-BPG, and then it gets to be the G3P that I need to make the sugars. I couldn't do that because oxygen was getting in the way. Oxygen was out competing me for the active site of the rubisco, and it was converting my rubip into this useless, just cutting it in half. It was useless to me. So instead, as a C4 plant, to overcome the idea that I'm holding my breath, oxygen's building up, photorespiration's happening, I don't have a storage of CO2, I'm going to store CO2 and make sure I'm good to go. So instead, they'll take the CO2 up here on the right, and they will use a different enzyme, Pep Carboxylase. Notice it's an ASE, so it's an enzyme. It's carboxyl, so it's adding a carboxyl group, and it's doing it to this molecule here, PEP. PEP is phosphoenol pyruvate. You see that in glycolysis, phosphoenol pyruvate. It's the step right before you make pyruvate in glycolysis, the second last step. Notice as well we have pyruvate down in the second cell becoming PEP. So that is the reverse of glycolysis. When you actually added the phosphate group in glycolysis, we're going to remove the phosphate group from PEP and to create pyruvate. So we're at the same molecules over and over again. And then the first molecule that we created was this 4-carbon oxaloacetate. We took this carbon dioxide up on the right, we squished it together with the 3-carbon PEP, phosphoenol pyruvate, and we made oxaloacetate, 4 carbons. Then that was converted to malate, and then we chopped the carbon dioxide off again. That left us with the pyruvate, and we were able to start again, add a phosphate. Now it's PEP again, and I can just keep doing that. And the goal here was that we would, in this way, transport so much carbon dioxide into the bundle sheath cell, which is impermeable to carbon dioxide, so it can't leak out, it's stuck there, that we would outcompete any oxygen that could possibly be there in that cell. And so that way, the rubisco, which is the enzyme that's supposed to take the rubip and attach it to the carbon dioxide, is able to work freely. So this is my normal Calvin cycle. So I want to stress again that every plant uses Rubisco. Every plant does the Calvin cycle normally, but these guys have an option where they can store the carbon dioxide first using Pep Carboxylase. That C3 plants don't have this option. So C3 plants, if it's too hot, they have to hold their breath, so they hold their water. These guys, well, that's fine. We'll just store carbon dioxide, and if we have to hold our breath, fine. We can still make sugars. Okay? So we've seen oxaloacetate, we've seen pyruvate, phospholinopyruvate, malate, all that stuff is already right here in this particular process. And what we're going to do is the reverse. So take a look now. You see oxaloacetate above becoming malate. And we go back to the Krebs cycle, we see malate becoming oxaloacetate. So now we take a look and we can see down here, we've got the malate becoming oxaloacetate. It's the reverse. Okay. So oxaloacetate then is this 4-carbon molecule. You can see it there. May as well label it as 4 carbons. 4-carbon molecule. I'm going to take my 2-carbon molecule acetyl group. So I'm going to crush those guys together, and I'm going to end up with my citrate. Because citrate is the first product here, this is why we also call this, citrate could be citric acid. It just depends on whether it has the actual H's attached to it or not. Because it is citrate is my first product, I can also call this the citric acid cycle. So Krebs is named after the researcher. Citric acid cycle is named after the first product. And the fact that if you look at the actual structure of the citrate, see this C-O-O? There's 1. That is a carboxyl group. Okay. Look here, there's another C-O-O. That's a carboxyl group. And then there's a third one. So that's why it's also called the tricarboxylic acid cycle. Okay, very annoying. You need to know all 3, that they are the exact same thing. Okay. So Krebs, named after the dude. Citric acid, named after the actual name of the first molecule. Tricarboxylic acid, named after the structure, the important structure of that first molecule. Okay. So what we're going to do first, after we've crunched those guys together, notice we've chopped off the coenzyme A. It'll be used later. You'll see it at the bottom of our little cycle here. I can either use it here, again, down below, or I can go and attach this to another acetyl group and make more acetyl-CoA. It just depends on where I am and what I want to do, right? So I'm going to go from citrate, and then I'm just going to make an isomer. I'm going to make isocitrate, and you can see the difference between those molecules. If you look carefully, you can see what's going to happen to them. It's really just a little rearranging. This, I've shown you here on the citrate, you just see this hydroxyl group move down, really, is what happens. On the next step, though, between isocitrate, this is where I cut off a carbon dioxide. And I take advantage of cutting off this carbon dioxide, and I steal electrons at the same time. There's going to be some kind of a question that asks you to put these molecules into their energy, like the main molecules, glucose, G3P, pyruvate, acetyl-CoA, and carbon dioxide. Well, you'll be asked to put them into their who has the highest energy. And think about, just follow the number of carbons, really. Carbons, and stored in between them is where we're getting our energy from, right? So if glucose has 6 of these carbons, clearly it's going to have more energy. G3P has more energy than pyruvate because you haven't stolen any electrons at G3P. That's the middle of glycolysis. You don't steal electrons till after that. So it's got more energy than the bottom. And then pyruvate has more energy than acetyl-CoA because it has 3 carbons, not 2. And of course, carbon dioxide only has 1 carbon. So it's not even, there isn't even a bond between another carbon there. Okay. So make sure that you understand that the highest energy would be the beginning molecule, the glucose, and the lowest possible energy would be CO2. So in other words, we've gotten, once we chop off the CO2, we have gotten all the benefit that we need out of that CO2. That's why it's worthless to us. There's nothing left to do with it. So out it goes. Okay. So citrate to isocitrate. Now I'm going to chop off a CO2 and steal electrons at the same time. Then I get to this alpha-ketoglutarate. That's that little symbol there is the Greek letter A, alpha. So it's alpha-ketoglutarate. We saw that in alpha helix coils. We saw that little prefix there. So it's the alpha version of ketoglutarate. Notice again, between alpha-ketoglutarate and the succinyl-CoA, The two C's, just like flaccid, where one makes a K sound and one makes the S sound, same thing here. So this is succinyl-CoA, and this would be succinate. Succinyl-CoA, succinate. So between alpha-ketoglutarate and succinyl-CoA, we have taken off our last carbon dioxide, we've stolen more electrons in the NADH, and we've shoved on the coenzyme A. So actually, that's the exact same process that just happened when we oxidized pyruvate. We chopped off a CO2, we stole an NADH, and we shoved on a CoA. The exact same thing. At the bottom here, though, there's something special that happens. The energy that we gain from breaking off the CoA in step 5 down here, between succinyl-CoA and succinate, that energy is enough for us to help phosphorylate a different version of an energy-carrying molecule called GDP. GDP just uses a different base. Instead of using adenine as your base, the A, adenine, you're going to use guanine instead. It's just the other nitrogenous base. Remember, we've got adenine, guanine, cytosine, and thymine, and then uracil if it's in RNA. So I'm just, rather than using the A, adenine, I'm using G, guanine. Slightly lower energy molecule, but it's enough that I can steal energy from the conversion of the succinyl-CoA to the succinate as that coenzyme A is being chopped off, and I can phosphorylate the GDP to GTP, but I don't leave it there. That's now enough energy that I can steal the phosphate off the GTP and make a real ATP, which is my goal. And so because the phosphate group started on an actual molecule, there's an enzyme that's going to do the ripping off, right? So that means that this is also considered substrate level phosphorylation because I'm stealing an existing phosphate group from an actual molecule, ripping it off with an enzyme and giving it to ADP. I'm not using the membrane to bounce and use ATP synthase to bounce electrons. Okay? So how many did I just make? 1 per cycle. Keep in mind, we are using 1 acetyl-CoA so far to run this, but I have 2 acetyl-CoAs. I had 1 glucose broken down into 2 pyruvates, which became 2 acetyl-CoAs, so this cycle runs twice. Okay, so per cycle, 1 ATP. Per glucose, this runs twice, I'd end up with 2 ATP. So you have to know what's your starting point. Are you starting with just a single pyruvate, or are you going all the way back to glucose, which means you double up all your numbers? Pay attention to the question so you give me the right numbers for the answer. Okay? Again, it's probably still seeming a little more complicated than it needs to be. I'm going to show you all the memory stuff in a second. So after I steal this energy and I'm able to phosphorylate the GTP, which then helps me phosphorylate ADP to ATP, I have one more chance to steal electrons from between succinate and fumarate. So I make an FADH2. FAD steals enough electrons to make FADH2. Fumarate gets converted to malate, and then between malate and oxaloacetate, I can steal my last little bit of electrons that are available. And it's important too because I need to get back to oxaloacetate so I can do this all over again. So just like in the Calvin cycle in photosynthesis, we used rubip, fixed it to the CO2. We had to come back around and make sure we had rubip to start again. Here we're doing the same thing. All of these, not only are we getting the advantage of picking up energy as we go, but we're also getting back to our starting point so we can start again. It's pretty brilliant, actually, that the end product is my beginning product. How clever is that? Like, I don't know, I think it's pretty clever. And we took advantage of stealing energy to get there. That's pretty brilliant. I didn't have enough room, so the reactants are at the top, and then you see there's an arrow in the middle there, and then the products are at the bottom, because I couldn't write it out side by side. So again, boxed down in green, we're following the carbons. Oxaloacetate becomes oxaloacetate. Brilliant. That's just like rubip being used and produced. Now oxaloacetate is being used and produced. So there's 4 carbons. Then the acetyl-CoA came in with 2. It got ripped up into the 2 carbon dioxides. So there's my 2 carbons there. And of course you got the CoA and the CoA. So that's in and out as well. You already know from photosynthesis, you just use an inorganic phosphate. You attach it to the ADP, you get yourself an ATP. Attach some electrons to an electron carrier like NAD, you get NADH. Attach them to FAD, you get FADH2. Done. So I have my 2 electron carriers. They're going to become very useful. I have my ATP, that's already useful because that was my end goal of cellular respiration anyway. And I now no longer have anything else to do with the actual glucose molecule. It's gone because those were the last 2 carbons that belonged to that glucose. That became those carbon dioxides. Done. So that's it. Now everything else from here on is going to be talking about what do we do with those electrons. I finished getting all the benefit I can out of the bonds between the original carbons. So let me show you now how you're going to memorize this and make it easy for you. I'm going to show you the full page for the Krebs cycle, just so you understand how to lay all the information out. So the oxidation of pyruvate is actually not, to be clear, is not part of the Krebs cycle, but it's a good place to start the conversation because we're talking about what happens with the pyruvate. So I boxed it out at the top there, but I just wanted to show you what the layout would be for what you want to write. And you can decide how that happens. And I'm going to give you, I guess, the next 10 minutes or so. But I'm going to actually go through and talk to you about what's happening in this little diagram. If you're setting it up, notice I've got the O right over top of the S and the C right over top of the SCOA down at the bottom, the succinyl-CoA at the bottom. And then you can see the fumarate and the alpha-ketoglutarate are opposite each other, M and I are opposite each other. So if you can kind of box it out like that for yourself, it's much easier to get your distances. Then notice what we do. After we take this acetyl-CoA at the top, what we're going to do is crunch it together with oxaloacetate, so O. It makes citrate. I convert it, it's just to an isocitrate. Then between this stage and this stage, I cut off a carbon dioxide. I steal electrons. Cut off a carbon dioxide, steal electrons, make ATP, so GTP to ATP, steal electrons, shift, steal electrons, and then I'm back to the beginning. So really what we have here is CIKSSFMO. So let me just shift that out the way. Come on. So this, if you look in the middle at the memory aids, CIKSSFMO. Take those first little letters and make yourself a sentence out of it. I used to have a really good one, and then one of my students, maybe 6 years ago, maybe 5, 6 years ago, I forget now. I think she's probably done university now, probably doing her master's. But she said that she used, "Can I keep Sally Snake for my own?" And that has stuck with me. I cannot remember what I used to use before, and I bet you it was super cool, but now that's what I'm stuck with. Can I keep Sally Snake for my own? Make up your own if you want, or do that one. And that's giving you the citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, oxaloacetate. Use this as your studying guide. Know what the words are, but use something like this to short form everything. It'll be way better for you to study off of this page than to go back to the complicated one, the big diagram, because you don't really care that, oh, okay, how exactly did citrate become isocitrate? Oh, the hydroxyl moved from carbon, blah, blah, blah. Who cares? You just need to know when it's an isomer, right? So from C to I is just an isomer shift, done. And the important thing is what came out. So we have between the isocitrate and the alpha-ketoglutarate on the right, carbon dioxide, steal electrons and make NADH. Next one, carbon dioxide, steal electrons, make NADH. Then at the bottom, GTP becomes ATP. Steal electrons and make an FADH2. Nothing, steal electrons, and I'm back at the beginning. So really we have shift, something, 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 nothing, something, and kind of give yourself like this little rhythmic kind of a thing to help yourself memorize it. What comes off? What comes off? And in general, you want to, again, we're following the carbons, so we know that the carbon dioxides have come off and we're back to acetyl-CoA. That's our major carbon stuff. In terms of energy, what did we just produce? Well, if you look around the entire thing, how many total NADHs came off? 2 on the right and 1 on the top left, right? So we have 3 NADHs produced. How many ATP came out of this? Just the one at the bottom. But really it was a GTP that then became an ATP. So what I produce directly from this cycle is 3 NADHs, 1 GTP, and this little FADH2 on the side. So other people have come up with things to say, well, it's 3N, a G, and an F, right? 3NGF. So 3 new goose feathers. That helps me to remember the products. And if you want to remember which one comes first, succinyl-CoA or the succinate, succinate is late. It's the second one. So there's these little memory devices that you can use. So can I keep Sally Snake for my own? 3 new goose feathers tells me what comes out of this. That's important. And then succinate is late. Tells me the order of those 2 S's. And then I've got my whole cycle. Remember as well, though, that this is a substrate level phosphorylation that happens. And the important products then, if I go right from the top of oxidation of pyruvate down to the Krebs cycle, I'm just going to zoom in here so you can see it bigger. From the oxidation of pyruvate, I chopped off a carbon dioxide to make my acetyl group, and I stole electrons and made NADH back then. So I have 1 acetyl-CoA to move into the Krebs cycle. From the Krebs cycle, I chop up the acetyl-CoA, I end up with 2 CO2s, 3 NADHs, 1 GTP that becomes an ATP, and 1 FADH2. And that's the terms of my carbons and my energy that I care about. Keep in mind, though, this is per pyruvate. How many pyruvates do you make from a glucose? 2. So this whole cycle, including the oxidation of pyruvate, will happen twice. So you're saying, if you're counting up everything here, you can see there's 3 carbon dioxides total between the 2 processes. So 3 carbon dioxides because pyruvate had 3 carbons, but glucose has 6 carbons, so this is half the step. So that's why I'm going to end up making 6 carbon dioxides eventually for my overall equation, right? Because this runs twice.